Hello guys and welcome back to Excel Share Academy. So yesterday we saw like how with the help of the proteins like importin and exportin, the protein molecules are exported and imported from cytoplasm and nucleoplasm, right? Now in today's lecture, we'll see how transport of RNAs are happening from nucleoplasm to cytoplasm. Okay, so there are many proteins which selectively transport from the cytoplasm to the nucleus. The RNAs are involved in protein synthesis are also exported from the nucleus to the cytoplasm. इसका मतलब क्या? Nucleus से हमने RNA, चाहे rRNA हो, tRNA हो, mRNA हो, everything needs to be transported to the cytoplasm. Okay, the export of tRNAs and mRNAs is mediated by specific exportins. Ribosomal RNAs are first associated with ribosomal proteins in the nucleolus, and nascent 40S, 60S, they are then separately transported to the cytoplasm by the exportin CRM1. Okay, so if you remember, Akshaya, we have gone through these proteins, right? Like different types of protein. So in the case of the RNAs, these are the proteins which is involved for the import and the export. Okay, so in the case of the export, it is CRM1, where 40S and 60S ribosomes are transported to the cytoplasm. Okay, now their export from the nucleus is mediated by nuclear export signals present on the ribosomal proteins. इसका मतलब क्या? Again, it is dependent on the signals which is present on these molecules. Now mRNA transport. So this we will be doing in detail. Okay, so mRNAs are exported by a distinct mechanism that does not involve the karyofuerins and it is independent of RAN. So what we are trying to say here is that like how in the case of protein transport, like yesterday we saw that we need RAN GTP, right? So if you remember, if it is in the nucleus, it is GTP. In the cytoplasm, it was GDP, right? And that's how we saw the export and import of the proteins. But in the case of your mRNAs, we don't want any, sorry, we don't want any types of importance or exportants, and it is also independent of RN, RAN GTP. Now, how exactly then this is being exported? So these proteins include distinct mRNA export complex, which is recruited to pre-mRNAs in the nucleus in concert with the completion of splicing and polyadenylation. So as we have studied during the transcription, so mRNA में आपके पास cap होता है, sorry, cap. It is your 5-guanine cap, 5-methyl guanine cap is present at the 5' cap, 5' end, right? And at the 3' end, you have got the poly A tail to which the poly A binding proteins are present, if you remember, right? Along with this, we have got another protein complex which is helping in the export of the mRNAs through the nuclear pores. Okay, so Akshay, if this point is clear?
Yes, ma'am.
Okay. Tanvi, recently you have joined. Any doubts? Like, so till now we have just started only. So what we have discussed is that in the case of the other RNAs and tRNAs, you need the karyofuerins proteins like importans and the exportins, right? But in the case of the mRNAs, it is not required. We also don't need RAN GTP. So what we are saying is that in the mRNA, we have got the 5' cap as well as 3' poly A tail is there, correct? This poly A tail is bound with your poly A binding proteins. So along with this, we have got certain extra proteins which is helping in the transport of the mRNA through the nuclear pores into the cytoplasm. Okay. Now, the directionality of the process is established by the RNA helicase localized to the cytoplasmic phase of the nuclear pore complex. Now, you must be wondering why we have RNA helicase. Any idea? And it is said that it is present in the cytoplasmic phase of the nuclear pore complex. इसका मतलब क्या? You have got the NPC over here. Okay, ये cytoplasm हो गया. ये आपका nucleus हो गया. Correct? This is your NPC. Now we are saying that RNA helicase is present at the cytoplasmic phase, यानी कि वो यहाँ कहीं present है. Now why it is important that RNA helicase should be present over here? Because helicase, it is going to remove the exporter complex, okay, because we don't want that exporter also to go there. Now this will release mRNA into the cytoplasm and prevents its transport back into the nucleus. Okay. Now we also have RNA helicase for another function is that जब आपका 5' cap है, mRNA है, and then 3' poly है, so there are chances that because of the complementarity base pairing, it might form a secondary structures. Correct. Now this will be hindrance in the transport. So we also need helicase so that this part of like secondary structure formation can be removed. Clear? Shall I go ahead? It is clear to you, Akshaya?
Yes, ma'am.
Now same thing. So we have got the 5' mRNA cap is here. Here poly A binding proteins are there. Now these are the additional proteins. Okay. Now these mRNAs are bound to the exporter complex, which will mediate their transport through the NPC. Now then again, we have got the helicase. So see, we have got the exporter complex, which is bound over here. Then we have got the helicase. Now this helicase will help in removing of these proteins, the exporter complex, and it will allow that it will go and get released into the cytoplasm. Clear? Simple, right? Oh, okay, sorry. What I made helicase was over this side. Has to be in the cytoplasmic phase. Correct. Clear? No confusion? Tanvi, is it clear? Okay.
Yes, ma'am.
Now, in contrast to this mRNAs, microRNAs, tRNAs, and rRNAs, which function in the cytoplasm, there are many non-coding RNAs, including the small nuclear RNAs, which is involved in pre-mRNA splicing, and the SNO RNAs, which is involved in RNA processing. They all function within the nucleus. So they remain in the nucleus, but there are certain RNAs, which needs to be transported from the nucleus to the cytoplasm, as already mentioned that it is done by the CRM1. Okay, so this point I'm emphasizing more and again because it is important. So transport of RNAs export in CRN1. Okay, now in the cytoplasm, the snRNAs associate with proteins to form the snRNPs, yani ki ribonuclear protein. Okay, small nuclear ribonuclear proteins. Sequences which is present on these proteins are then recognized by the important called SNRPotin, which mediates their transport back to the nucleus. So एक बार काम होने के बाद it needs to come back. So that will mediate its transport back to the nucleus. See again. So this is the transport of snRNAs between the nucleus and the cytoplasm. Okay. So you have got the snRNPs. These proteins are there. They go and bind to the SNRPotin. And then here inside the nucleus, they get released and protein also comes back. Now you have got the small nuclear RNAs. CRM1 क्या है आपका? Export करने वाला protein, if you remember, right? So this CRM will be bind to this RNA. Can you see in the diagram? This is your rRNA. Okay, RNA. CRM1 will go and bind to it. So this will help in the transport from nucleus to the cytoplasm. Okay, अब क्या हुआ? आपका RNA बाहर आ गया और ये प्रोटीन भी आपका एक्सपोर्टिंग प्रोटीन भी बाहर है. So that will be again taken back inside, if you remember in the previous diagram. So with the help of the RAN GDP, it will be taken inside the exporting proteins. ठीक है? Now these are the protein binding. This will go and bind to these snRNPs. Now entire thing will be taken inside. अभी हमने क्या किया? SNRPotin क्या है? एक तरह का important है. Correct? So this will bind over here and will come inside. So this particular thing is now present in the nucleus for its function. Clear guys? अभी यहाँ पे एक सवाल ये आना चाहिए कि instead of sending the snRNA into the cytoplasm, इससे अच्छा क्या होता है? डायरेक्टली आप प्रोटीन बाइंडिंग, ये प्रोटीन्स को आप अंदर ले आते. करेक्ट? उसको बाहर भेज के फिर वापस अंदर लाना, instead of doing that, it was easy that नर प्रोटीन आपका इस प्रोटीन को डायरेक्टली अंदर ले आता है न्यूक्लियस में, यहाँ आके एसोसिएट हो जाता है. So why in the nature this mechanism has been accepted and not the other, which just now I have said? So what will be your reasons? Yes, Akshaya.
Might be like these proteins have to be synthesized inside, and it needs to be recycled. So, in terms of
So the protein synthesis happens in the cytoplasm. What I'm saying is, instead of this RNA to come into the cytoplasm, binds to the protein, and then again recruited inside, instead of that, what the nature must have done is take these proteins and bring it over here, then associate with the RNA to form the snRNP. ऐसा क्यों नहीं किया? तन्वी, क्या लगता है क्या रीजन होना चाहिए? सोचो. Okay, do you remember where we use snRNAs or snRNPs? So we use in the splicing or in the spliceosome, if you remember. हाँ, RNA splicing में हमने पढ़ा है ना, U1, U2, U3, 4, 5, 6, they together. They're nothing but your snRNPs. Okay, small nuclear ribonuclear proteins they are. Okay. Now the answer is that, see, the protein will have its activity when it is bind to the specific target. Okay, so the target was required so that, see, how should I explain? Okay, so when this RNA was outside in the cytoplasm, these proteins were required, right? तभी इनका synthesis होता है. So you need certain expression, you need certain stimulus, right? You need certain stimulus so that the proteins are getting synthesized. Now, when this RNA was there, so there was a signal to the cell that these proteins need to be synthesized. So that time they were synthesized in the cytoplasm, and now when they have got their snRNA, they go and bind to that. And that's how, when it becomes a functional complex, it is then recruited inside the nucleus. Now it is clear, understood? What you have to remember is, yes, Akshaya?
can you repeat again?
Yes, okay. So first thing is that our cell, okay, it does not synthesize anything without any reason, okay, because it needs energy either in the form of ATP or GTP. Just imagine how, for example, you are there, you know that I have got only 1,000 rupees and I should use very cautiously. ऐसे मुझे पूरा महीना चलाना है, ठीक है? I don't have much currency. So you will be thinking, right? When the requirement is there, then only you're going to buy. Correct? Leave the today's scenario that we so just buy for no reason because we know that we have got money. Just imagine you have only 1,000 rupees or 1,080 are there. So you are not going to synthesize until it is required. Okay. So now when the snRNAs is brought into the cytoplasm, so it was a stimulus. It was a signal to the cell that this needs its protein components to become a complete functional spliceosome so that the splicing can be done, so that the transcription, after transcription, whatever RNAs are present can synthesize functional mRNA so that I can have a further response. तो उसके लिए क्या करूंगी मैं, पहले जाके उनको बोलूंगी, see I have got the RNA, why don't you start making the required proteins. Okay, so this snRNAs now it is going to get complex with the required proteins. So back में क्या हुआ, these proteins were translated. It is not that they're already present in the cytoplasm. No, it is not. When the signal was there that snRNAs are there, now I need those associated proteins, please synthesize. So cell is going to synthesize. Now it is present in the cytoplasm. So it will come and it will bind, and entire functional spliceosome will be then recruited inside for your RNA splicing. Abhi samjha?
Yes, ma'am.
Okay. Wow. Okay, so that's all guys. Chota sa hi topic tha. I couldn't prepare much. I'll just stop here.